I'll explain a bit of what you'll be doing in the genetics lab if you haven't figured out the details already. Again, the idea is to try things out and see what type of pattern you get. Real world is not an infinite number of experiments similar to what Mendel was doing. Instead, we have smaller samples, and also the sets of parents we have are not typically standardized the way they were for Mendel's experiments. So we'll see a little bit of both how things work modeling something like what Mendel had, and some of the reasons why the real world patterns actually are somewhat different. So, for example, we can think about what would happen if we were to cross two individuals. One of them is homozygous for the dominant version of this G gene, and the other homozygous for the recessive. So what you want to do is get 20 each of four things. What I've done is just use pieces of paper here. So we've got get them right set up here, capital G, capital R, and lowercase g and r for my four piles of paper over here. And then I'll use those to represent the alleles, the individual copies of the gene in the parents, and then draw from the piles to see what actual gametes I get, and thus what the offspring would be like. So for example here, again, we're crossing the parents, write that a little bigger. Big Okay, so we've got one parent that has two of the big G alleles of the gene. So no matter what happens in meiosis, each gamete's going to have the big G because that's all that's in the parent. Same for this one. All the parent has is the little g, so the gametes are always going to get little g. Okay, so big G from one parent, little g from the other. Well, it's going to get one of each. Not too hard to figure that out. And you can follow the pattern through the rest of these squares. To model this with my pieces here, what I can do is take one piece without looking at it from a pile that's got all big G's, and one from, make sure I'm grabbing the right pile here, from the pile that's all little G's. And guess what? Without looking at the piece of paper, I know what I got. One big G and one little G. No, I'm not psychic. Okay. It's pretty obvious what sort of combination we're going to get if one parent has all one thing and the other parent has all one thing as well. Okay, what gets more complicated and somewhat more interesting is if we have a few more variations here. Get it back in the right spot. Okay, that's got a little bigger. So again, what you will do is figure out what will happen. Let's see, that's the one we just did. This is what the top. So this is your chart where you'll fill in what happens with each combination. Again, this time we've got parents that have one each. Each parent has one of the uppercase dominant version and one of the lowercase recessive version. So Now, either parent could make a gamete with the dominant or the recessive, and any one gamete could end up pairing up with any other gamete. So we'll get more variety as you fill in this chart. Hopefully you've figured out how that works by now. Again, there's detailed examples up at the beginning of the lab, and feel free to email me questions if you're still trying to figure anything out here. Okay, so that's what we expect to be the pattern, assuming that we had lots and lots of individuals to test getting large numbers here. You probably don't want to try modeling this 100, 200 times, something like that. So we'll see what happens if we have a somewhat smaller set. And so what you'll do to model this is in our next table down here, so what you'll do is take 10 of your big G things, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 10 of your little G things, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, mix them together so you don't know what you're drawing out there. Okay, so now I have my mixed pile, and without looking, I will draw out 2 pieces of paper and see what I got. And I have 2 big G's. Okay, so I can put 1 tally mark in the 2 big G's pile. Okay, or square rather, not pile. Put those back in the pile. Mix around again. Get 2 pieces of paper without looking at what letter they are. This time they were both little g. Okay, so I've got one of those. And so what you'll do is do that until 12 times. See how many of each of these you actually get. And then you can write that as a ratio. For example, let's say you ended up with, all right, let's say 2, 8, and 2. That adds to 12. Okay. Then your ratio, or you could just write as 2 to 8 to 2, or well, all multiples of 2, so that's the same as 1 to 4 to 1 for your ratio. And that's whatever you get. Again, you just calculate what, in theory, you should expect. And if you got 1 to 4 to 1, well, it wasn't quite what the theoretical pattern would be, but then again, just 12 samples. Not too surprising that that doesn't necessarily get you the exact expected ratio. Comparing your ratio to total for class. Now, I posted last year's totals. So that file's up on Blackboard, and you can use those numbers. Or if you want to wait, you can send me what you got for these, and I can kind of see what everybody got, whichever's more convenient, probably looking at last year's class. But you go ahead and not wait for everybody else to turn in theirs as a comparison. Of course, the point comparing to the whole class's values, that's a bigger sample. Or if you don't have anything better to do, you could sit around and do lots more than 12 times and see what pattern you get from drawing out many more times and how that ratio compares to what you got when you just did 12. That would be another way of doing that part of the lab. Again, the point is comparing what happens with a relatively small sample versus a relatively big sample. Okay,